and we're going to be hearing now from Dr. Kim Vanuytsel. Dr. Vanuytsel is a stem cell biologist and assistant professor of medicine, also in the Center for Regenerative Medicine at Boston University, with expertise in hematopoietic biology. And Dr. Vanuytsel has focused her lab on developing tools and resources that help us understand important concepts in hematopoietic development, with the goal of translating this knowledge through iPS cells and hematopoietic stem cells into better treatments for patients, and with a special focus on patients with sickle cell disease. Through the magic of technology, we're going to be hearing from Dr. Vanuytsel remotely, and her talk is entitled "A Sickle Cell Disease-Specific Induced Pluripotent Stem Cell Platform as a Versatile Tool to Assess Therapeutic Options Across Diverse Sickle Cell Disease Patient Populations." And I think I am starting the talk. Let's see here.
Good afternoon. I'm Kim Vanuytsel. I'm an assistant professor in the Center for Regenerative Medicine, as well as in the section of hematology and medical oncology. I'm very excited to share with you our work on the validation of a sickle cell disease-specific iPSC platform as a versatile preclinical screening tool. But before I dive into our platform, I'd like to start with a little introduction so that we're all on the same page. Sickle cell disease results from a point mutation in the beta-globin gene, which renders hemoglobin prone to polymerization. So this polymerization makes the red blood cells rigid and easy to break, resulting in anemia, as well as the sickling of red blood cells, leading to obstruction of blood flow, resulting in painful crises and a host of downstream effects, severely impacting both the lifespan and the quality of life for sickle cell patients. The main therapeutic strategy is fetal hemoglobin induction. And fetal globin or fetal hemoglobin, it's a globin that's typically around prenatally, and it's perfectly suitable to carry around your oxygen, while at the same time it interferes with the polymerization process. So there's a few strategies. So we have disease-modifying drugs, where hydroxyurea is the mainstay drug for sickle cell disease for the past few decades, and it helps in some cases, but there's also a number of patients who do not respond to this therapy. And then on the other hand, you have a more recent development thanks to the recent advances in gene therapy. It has now become a real possibility to treat sickle cell patients with their own therapeutically modified stem cells. And this is a curative approach. And one of the main approaches here is, again, fetal globin upregulation. So we're very proud here at BMC to be able to offer these therapies to our patients. And in fact, our lab works with some of these gene therapy samples to try to further improve the efficacy, the safety, and the accessibility to these treatments. Now, while these treatments are really transformative, there exists significant barriers still for most patients to gain access, and one of them is eligibility, but the most important one is the cost. So that means that as a field, we need to keep investing in the development of treatments that can alleviate symptoms for patients globally who might not be eligible or just reside in lower resource settings. And this is where our sickle cell disease platform comes in. So thanks to our collaboration with our colleagues of the Center of Excellence in Sickle Cell Disease, we have previously established this comprehensive and ethnically diverse library of sickle cell disease-specific induced pluripotent stem cells. Now, I just told you that sickle cell disease results from a point mutation, the same point mutation in all patients in the beta-globin gene. However, the genetic background in which this occurs to some degree dictates the severity of the disease course as well as the response to treatment. Now, all of that variation is here captured in our iPSC platform. So that means from a range of patients globally, we've managed to reprogram their cells into induced pluripotent stem cells, which means that we now have an unlimited resource of cells corresponding to these patients that we can further differentiate in whichever cell type we like. And in our case, we're obviously interested in the erythroid lineage because that's where the disease manifests itself. So on the right, you can see a hypothetical example of how we think this could work. So we have patient-specific erythroid cells, which we can then treat, for instance, with hydroxyurea or HU here, and see the differential response to treatment, where some patients respond well and others not so much in terms of HbF induction. And then, for instance, we can test compound X, Y, and Z. And perhaps we find that compound C could be a solution for this patient number 2 here, where hydroxyurea just doesn't cause fetal globin induction. So that's kind of the idea behind this platform. And the reason why we think this is important is because historically, when new drugs are developed, often a drug mechanism is discovered in a cell line, maybe tested in another cell line, and then it goes into clinical trials eventually, where it meets a real patient population, and it turns out that this is effective for 20% of the people, but not for the other 80% of the patients. So we're hoping to improve on that by adding some of this genetic diversity upfront in a more preclinical stage. All right, so to make this happen, we first spent a lot of time trying to make the best possible erythroid cells. We were successful at making in vitro-derived erythroid cells here. And in the process of characterizing the cells that we were making in our dishes, we also learned that these iPSC-derived erythroid cells resemble their postnatal counterparts in many ways. So they have a very similar erythroid differentiation trajectory, for instance, as compared to when you take cord blood cells and differentiate them to the erythroid lineage. And we judged this based on marker expression, as you can see in these flow plots. And also we tracked the different progenitor stages using right Giemsa staining, seeing that everything was on course. And as you can see from the pellets here above, these cells turn nicely red, as you would expect. We also found in our transcriptomic analysis of the cells that we were making that the dynamic developmental processes that control erythroid maturation postnatally are also at play in our in vitro-derived cells as they mature. And then, moreover, when our cells were used as a control in this large proteomic study, we found an encouraging degree of overlap between our in vitro-derived cells and the postnatal cells, red blood cells, that are present in our bodies. So really encouraging. The main difference that I do want to highlight here is that in contrast to postnatal erythroid cells, which predominantly express adult globins, our iPSC-derived erythroid cells predominantly express fetal globins. And now you can imagine that this limits certain aspects of disease modeling, especially the more advanced ones like the sickling of the red blood cells. That's not something we can model in our system. However, we made sure to validate that the predominance of fetal globin expression is not an obstacle for fetal globin induction screening. And I'll show you right here. So we're looking at iPSC-derived erythroid cells. You see they're nice and red. And if we then look at them by flow cytometry, we can see that here on the y-axis, I have CD235A or glycophorin, which is a pan-erythroid marker. And you can see that the majority of our cells are expressing this pan-erythroid marker. And then on the x-axis, we have HbF50, so fetal globin expression. And you see when we treat the cells with water, which is a control condition, we already find fetal globin expression, as I just mentioned. However, when we treat with hydroxyurea, a very well-known fetal globin inducer and the main drug used in sickle cell disease, you see a clear shift into the right quadrant, so meaning a shift towards HbF positiveness. And we can visualize the same thing for these glycophorin A positive cells here in this histogram below. Again, you clearly see the shift between the hydroxyurea treated conditions versus the water, the control. And then we can further quantify this by taking the mean fluorescence intensity and expressing this as bar graphs. And this is how I'll show you most of the data in this presentation. But basically, this first proof of concept, if you will, fetal globin induction in our iPSC-derived system was very exciting, but we were, of course, very interested to understand how that was different between different backgrounds for a certain drug. So we went ahead, and here we are looking at different patient lines. So we made a selection of sickle cell disease specific IPS lines representing the different haplotypes. And we also included BO6, which is our healthy control cell line. And then we took all these cell lines and we started to differentiate them with our optimized protocol. So we first make hematopoietic stem and progenitor cells from these cells. And then we further specify these towards the erythroid lineage. So then shortly after we induce this erythroid specification, we treat the cells with fetal globin inducers. In this case, I'm going to show you hydroxyurea, the main drug, and RN1, which is an LSD1 inhibitor, just for a variety of drugs. And then we typically read them out a few days later. So I'm showing you a lot of data, but I'll go through it in more detail. But basically what we saw is that our iPSC-derived erythroid cells reflect the diversity in hydroxyurea-induced fetal globin induction seen in the sickle cell disease patient population. So basically what we found is a differential response to fetal globin-inducing agents, both within and across the sickle cell patient backgrounds. But just to go through this more detailed, I'll start on the left with the healthy control cells, or BU6. And you can see compared to the control, which is water in this case, there's a clear fetal globin induction as shown by mean fluorescence intensity here in BU6. And also for RN1, we see a very clear fetal globin induction when we treat with this compound. So that's our healthy control cell line. And then on the right, we have all the sickle cell backgrounds we're looking into. And you can see that for some of these lines, like SA170 corresponding to an Arab Indian haplotype, for instance, you can see that there's a similar induction upon addition of hydroxyurea and RN1. Some other ones like SS24, there's maybe a trend not significant, but we can also see that for some of these cell lines like SS100 and BRSP31, nothing happens. There doesn't seem to be a response upon adding these fetal globin inducers. And this is true across many repeats. So I want to highlight that this data is representative of 3 to 9 biological repeats, meaning that we take these cell lines and do individual differentiations, week-long differentiations with these cell lines, and we consistently see no fetal globin induction for some of these cell lines. Very similar to how in a patient population, some patients respond to hydroxyurea and others do not. Right, so to make sure that we're not just like underdosing our cells, we also increased the dose of hydroxyurea. So the one we're working with here is 50 millimolar, and this is like widely accepted in the field in lots of in vitro studies. This is what people use and see in cell lines. They see fetal globin induction, so that's what we did. But then we decided to also add a higher dose, 200 millimolar, and see what happens then. And basically, in conclusion, we find that the absence of fetal globin induction response upon hydroxyurea treatment is not due to insufficient dosing. So again, a lot of data, but basically what we're looking at is even if we add a higher dose, it doesn't really change the results. So cell lines or iPS cell lines that responded to 50 millimolar kind of have a similar response or fetal globin induction even at a higher dose, although the results get a bit more variable at this high dose. And then if we look at SS100 and BRSP31, again, if we see that these cell lines did not respond at the regular 50 millimolar dose, 200 millimolar doesn't change that neither. So it's not a matter of just underdosing the cells. And interestingly, we also found that for one of these cell lines, like SS100, we did have the clinical history of this patient, and we know that this patient, despite being on hydroxyurea at the maximally tolerated dose for years and years, their fetal globin just does not increase. And this is very similar to the results that we find using the cell line or a cell line representing this patient background in vitro. So basically, these cells can recapitulate a treatment response corresponding to a clinical outcome in a sickle cell patient. Now, the nice thing about our platform is that we have a unlimited amount of cells from this patient background. So we can start looking if we can find alternatives that could induce fetal globin induction, whereas hydroxyurea does not. So that's what we try to do here. So we're expanding our fetal hemoglobin inducers pool. I already showed you results for RN1 and hydroxyurea, but we're adding now pomalidomide, decitabine, and SR18292. All compounds that in cell lines in the literature have been described to increase fetal globin expression or reactivate fetal hemoglobin. And first, we're looking again in our healthy control BU6. So in line with the results I just showed you, when we add hydroxyurea, there's a clear response, clear fetal globin induction. And then the other ones have DMSO as a control, but you can see pomalidomide, decitabine, and RN1 increase fetal globin. This SR compound, not so much in this case, but when we then look at our SS100 patient cell line, we find that, as I showed you before, hydroxyurea doesn't really make the cells increase their fetal hemoglobin expression. And then neither does pomalidomide or decitabine. RN1 also doesn't seem to do anything beneficial there, but this SR18292 does seem to cause a significant increase in fetal globin expression. And this compound is a PGC1 alpha agonist. And the reason why we added this to our pool is because our colleague in the Center of Excellence for Sickle Cell Disease, Dr. Shuang Shui, has described last year how PGC1 alpha agonism induces fetal hemoglobin and exerts anti-sickling effects in sickle cell disease. So we thought it was an interesting compound to test in this scenario, and it could actually be beneficial for this particular patient. And we're currently doing dose curves to understand what a good dose would be in this context. So all possibilities of this platform. The last vignette I want to leave you with, just to illustrate the versatility of this platform, is this one right here. So because we have an unlimited amount of patient material, we can do these controlled and repeated experiments, which is kind of harder when you work with primary patient samples. So here, for instance, I'm showing you how we discovered that some drugs work differently. They have a different, well, mechanism, of course, but also target different progenitor populations. So early on, we learned that if we add RN1 at the same time as we add other drugs on day 15, right after erythroid specification, that just doesn't work out very well because of what I'll show you next. So typically, when we induce our erythroid specification, we add the fetal globin inducers right after, and this doesn't affect the maturation of the cells for many of the compounds we tested. So I'm showing you in the upper row here the results for DMSO, which is a control, but basically at day 15, when we add the drug, you can see on the y-axis that there's some glycophorin A specification already, so specification of erythroid cells. And on the x-axis, I have BAN3, which is an erythroid maturation marker. And you can see that over time, from day 15 to 18 to 20, there's an increase in BAN3 expression. So these cells keep maturing and keep doing what they are supposed to do. However, when we add RN1 at the exact same time, when the cells look the same and some cells have started to specify towards the erythroid lineage based on their glycophorin A expression, we find that only those who have made it there already before we start dosing can continue to mature, and they will further mature. However, there seems to be a complete block in further specification towards the erythroid lineage, as you can see in these graphs. So it's very interesting that we can use this model system to really dissect which progenitor stages are affected and how a drug works more mechanistically. All right, so in summary, by showing you our results with our sickle cell disease iPSC platform, I hope I told you that the predominance of fetal globin expression is not an obstacle for fetal globin induction screening, with fetal globin induction being the main therapeutic strategy. I also showed you that our iPSC-derived erythroid cells reflect what is seen in the sickle cell patient population overall in terms of their response to hydroxyurea, where some patients' backgrounds seem to respond and others do not. We also talked about how these cells can recapitulate a treatment response corresponding to clinical outcomes in sickle cell disease patients. So our SS100 sample does the same as what is seen in clinic. And this then allows for looking for alternatives and patient-specific recommendations. I also touched briefly upon how our unlimited supply of cells provides a window into drug kinetics, the precise progenitor stages targeted by a drug, and drug toxicity. So I didn't talk about drug toxicity because of, limitations of time here, but you can even think about making from the same patient-specific sources hepatocytes and looking at toxicity markers. Really, there's no limits to what we can do there. And in general, I would say that our sickle cell disease iPSC platform expands the preclinical toolbox, adding genetic diversity as an important and often overlooked component. So, of course, like any tool or model system, it has advantages and limitations, so a combination of approaches remains advisable, but I think it's a nice addition to the toolbox. All right, so all that rests me is to thank the people involved in this work, and I also want to say that I'm grateful for the support that we received to pursue this work, as well as other lines of research established in my lab, including CTSI pilot funding. So thank you for your attention. Apologies for not being able to make it in person, but Braden and Maddie should be somewhere around. So if you have any questions, you can try to look for them or email me if you want to know more. Thank you.